So the problem right now is that our oceans are quickly deteriorating due to human-caused pressures, largely being climate change, and specifically the warming of our oceans are causing harmful algal blooms to become more frequent and more potent. And so now harmful algal blooms are dangerous because there are certain algal species that can produce toxins that are then released into the water. So now this can be dangerous towards human health because it can infiltrate the food chain. So by the time it makes its way onto our plate in the form of fish, mussels, clams, or oysters, it can lead to large medical complications such as memory loss, seizures, and even death. This toxin is additionally scary because we cannot taste it, we cannot see it, cannot smell it, and when we cook our seafood, it doesn't damage the toxin. So to address this challenge, we looked towards synthetic biology, specifically towards CRISPR-Cas systems.
Can we develop a quick, easy, and cheap way for the CRISPR sensors to detect harmful algal blooms in the ocean? And to do that in a way that doesn't really require sophisticated laboratories, rather than the way we've been doing it, which is you kind of take these samples, bring them back to the lab, you have to do the analysis, and it's a huge time sink. There's like a week lag between when you take the sample and when you actually have the results. So imagine if you're a beachgoer out there, eating mussels off the beach because you're camping. An alert coming out a few days later, that's too late already, right? And these toxins have effects beyond just human health. They decimate whale populations and bird populations as well. And so having the answer within minutes there is really critical at devising a contingency plan for dealing with how the ecosystem is going to respond to this algal bloom, as well as making sure humans are safe.
CRISPR's programmability is so exciting, and it's the beauty of it, because we can repurpose that process towards other harmful species. And we've already done this actually with Vibrio species, which is a bacteria that is harmful and deadly towards humans, as well as largely been impacting oyster reefs. This summer, I hope to actually at the Duke Marine Lab extend and expand upon this project towards creating more sensors, specifically towards harmful pathogens, those that are ravaging coral reefs and seagrass beds, and possibly even extend this project further towards creating a sensor that can characterize stress in dolphins by focusing on their DNA methylation.
This is the first foray of our CRISPR platform into looking at environmental DNA sequences, and we're targeting ocean health. We've developed these sensors, we've verified these sensors work, and now we are ready to actually test them with real ocean samples, deploy them out in the ocean, and get it working in a real-world scenario.